Enzymes are a type of protein catalyst. They speed up reactions by lowering the amount of activation energy needed for that reaction. Enzymes are extremely important because they are responsible for every reaction that takes place in our bodies. Without enzymes, we would be unable to perform the necessary functions of life. Enzymes are mainly tertiary-level globular proteins. Enzymes are very specific about what they can catalyze. Lactase, for example, will not catalyze anything other than the reaction that causes the digestion of lactose. If you were looking at a lock and key, the enzyme would be the lock and the substrate, or the substance the enzyme is catalyzing, would be the key. For example, an enzyme is what generates the reaction that breaks up the disaccharide maltose into two glucose molecules. In this case, maltose is the substrate, the enzyme is the enzyme obviously, and the active site is where the key fits into the lock. When maltose enters the active site, the enzyme lowers the activation energy needed for the hydrolysis of maltose, and it breaks into two glucose molecules. When you look closely at the active site, it is full of R groups that react with the R groups on the substrate to cause the reaction to occur. Enzymes are usually made up of a protein and a cofactor, or non-protein. Because enzymes are so specifically structured, factors such as extreme heat or pH imbalance can cause them to stop functioning. Enzymes are involved in, but not affected by, chemical reactions in the body. Enzymes lower the activation energy, or the amount of energy needed, to start a reaction. The function of an enzyme is determined by its protein structure. The way the R groups are arranged on the active site determines which substrate will fit into the enzyme to react. Enzymes lower activation energy by temporarily combining with the substrate. When the substrate and enzyme combine, it is called the enzyme-substrate complex. Since enzymes are so particular in which substrate they can combine with, the enzyme-substrate complex has a very precise fit called an induced fit. Enzymes bring molecules close together and align them perfectly to begin reacting. Lactase, as discussed earlier, is an enzyme that breaks up the disaccharide lactose into the monosaccharides glucose and galactose. This reaction is necessary for the digestion of lactose. Lactase is usually naturally produced by the body, but some people suffer from a condition called lactose intolerance. People who are lactose intolerant do not naturally produce enough, or sometimes any, lactase. Therefore, they must either avoid dairy products that contain lactose or use an artificial enzyme supplement that digests lactose. Lactase is made up of two active sites. One site is structured to hydrolyze galactose and other similarly structured molecules, and the other site is structured to hydrolyze glucose and other similarly structured molecules. The amino acid sequence of the enzyme lactase is 1,927 amino acids long.